Hi everyone, thank you for coming. We'll start in around 8 minutes. In the meantime, I will post in the chat the paper link and I will put up also the presentation so you can check it out in the meantime. And our guest speaker's website, which is really also interesting to check. So there's information. Hi, Nozomi. Sorry, I was chatting and I was in the wrong screen, so I didn't see you. How are you?
No problem. Good. I just joined. Can you hear me?
Yeah, perfectly. Thank you.
Awesome.
It's like you switch like from screen to copy the links, and there it is. Should be working for everyone. Please let me know if it doesn't, then I'll check out the settings again, but I think it should be fine. How's your day so far? I'm very happy today because Portugal just won. 6
That's
to
exciting.
1, like in the soccer World Cup.
Yeah, that's
So
amazing.
yeah, it's really amazing. And yeah, so usually it's like watching Portuguese game for me and my family is like very stressful, like very emotional and very stressful because it's like if they are losing or like almost, or it's usually it's horrible, but until now it's pretty relaxing, the World Cup. So
Nice. Well, congratulations. That's exciting.
well, thank you. I was actually rooting for for teams that were not usually would come for, like Japan did so well in the group, and then South Korea, and the US didn't. US is not like a big soccer nation, so I was happy, but then they kind of got kicked out lately. Like, well, that was sad, but
Yeah.
I'm sorry, it's probably not interesting, but we have some time.
I wish I were, a normal person and could tell you more about what I think about soccer. I'm married to a Brit actually, except he doesn't watch football. So,
Yeah, I only watch
um,
these type of cups, World Cup or European Cup. It's like I grew up with that, so even my great grandmother, she used to watch those games.
yes, my
So
family's definitely into it, and I'm just not very good at keeping up with any sort of sport. So,
yeah, other than that, I wish, like, I'm all for Olympics and stuff, but I kind of never get around to actually follow it then. I watched actually a really good movie recently that was about two Syrian sisters that escaped and then found asylum in Germany. And they were swimmers. They were training actually for being qualified in the Olympics in Syria, and I think they were qualified with that time. And then one of the sisters ended up swimming for the refugee team. Like the Olympics decided to make a refugee team that don't have a home anymore. And it was such a beautiful movie. But then one sister is actually being arrested because she ended up going back and helping refugees that, a lot of them die in the ocean to help. And then they claimed she would she was for an organization that helps these refugees survive. And the EU is being very shady, like just leaving them out in the ocean, and they don't want people really to help. She was arrested for it. I don't know what's. But anyways, it's a very beautiful story in
I think
general.
I think I saw the trailer on Netflix. Is that where you saw it, or?
Yeah, exactly. So
Mm,
yeah, that was good.
yeah, I'll check it out.
That's all. He's already. Thanks for your question, and that's exactly what we will be discussing. Thanks for posting that question already, and then we will for sure get around to it once we get to present it, like when Nozomi gets to present on the talk and then the Q&A. So yeah, thank you. And yeah, let me been chatting. Let me share on Twitter that we are about to start. And if you feel like this is something people would like that feel free to share it. It's a public room, so anyone can just come, and we welcome people to join us. Um, and yeah, we will start in around 3 minutes. So would be will be really interesting. Really looking forward to this. And Nozomi, of course, thank you for taking the time, to make that count and come here. I know it's probably, like you have probably a lot of things to do, so we really appreciate.
No problem. Thanks for the invite.
Yeah, and if you like discussions like this, follow the club. We also have actually a Spotify and a YouTube account, and I have to get into the habit of sharing those links too. But yeah, we will have a little bit of a break around the holidays, and then we'll start fresh again in January. So we have a few more talks before the holidays, but not too many, and then we'll restart again in January. I'll have to take time to organize the program. So, but it will be for sure exciting, and a bunch of guest speakers already promised um, to come back. So yeah, if you missed talks, you will for sure get opportunity, at least for some, to speak with them um, in the new year. And hi, Victoria, meet Nozumi. And yeah, Nozumi, meet Victoria. She's a wonderful, wonderful moderator, and she does this really interesting. She started doing this really interesting beginning interview part. It's totally her idea, and it adds so much to a discussion. I was like the traditional conference style room that I did, and Victoria added this more personal um, interview part, which is always very interesting. So hi, Victoria.
Thank you. Thank you for that very kind introduction and generous. Nice to see you. Nice to see you, Nizomi and Katarina. Sorry, I can't see my phone very well if I pronounce anyone's name wrong. Yeah, really looking forward to this talk today, and was so interested in reading your bio and your information. It was really beautiful, the words that you used in there. Katarina, I don't know if you've already mentioned that, but that acknowledgement of inclusivity it's just so appreciated and important, and it's wonderful that you that you took the time and energy to say the things that you say. So thank you.
Thank you for that. Thanks for noticing.
Yeah, no, I didn't, and it's right on time. So that was the perfect start for, for this. So thank you, Victoria. And
Go team.
exactly, that was as if we have had practiced it, but it was spontaneous. Um, yeah, so welcome everyone to Science Society, and of course, a special welcome to you, Nozomi. And before we start, let me introduce you to the audience so they know a little bit about you. Um, so, um, Dr. Ando. She did her bachelor in science at the Massachusetts Institute of Technology and her PhD at Cornell University. And then later on, she did a postdoc at the MIT. And Nozomi was born in Denver, and she grew up in the Boston area. And she the decision why I thought that was really cool. Also, why she really liked the she did a physics major at MIT and a music performance minor, which is also really interesting because I kind of feel that or realize, maybe just biased, but that a lot of our guest speakers here kind of have a passion for both, for music and science, which is really interesting. And she was drawn to the spaceship-like feel of the Cornell high-energy synchrotron source, CHESS, and went to Cornell University for a PhD in physics. And she was in Saul Gruner's lab, where she made her own diamond cells for high-pressure X-ray scattering studies, and then went on as a postdoc in Kathy Drennan's lab at MIT, and there she developed a fascination for metalloenzymes. And then later in 2014, she was appointed as assistant professor of chemistry at Princeton University and started a research program that combines X-ray physics and structural enzymology. And in 2018, the Ando lab moved to Cornell University, where she joined the faculty of chemistry and chemical biology. And as I said, outside the lab, she is a classically trained soprano. She takes her hobby quite seriously, and she also enjoys observing and naming wild woodland animals. And she that's interesting because I kind of feel the same, that the kitchen is her lab outside of the lab, and I always think if you can follow a lab recipe, then you can also cook. Because some people, like students, tell me, I can't really cook. I said, if you can follow a PCR recipe, you can follow like a regular recipe. But
Right.
yeah, so where she bakes bread and she makes miso and various Japanese recipes or cooking art, um, which is really interesting. And also, her current tools of choice are Japanese donabe pots made of ancient clay, and the clay is sourced from regions that used to form the bed lake of Biwa in prehistoric times and is thought to contain lots of fossilized microbes, which is also really interesting because it kind of closes the circle there.
It does, yeah.
Okay, well, this is such a pleasure and such a treat to meet you here and to get to know you. And Victoria, the stage is yours. Thank you.
Thank you so much. Can we just talk for a long time? I had so many questions coming up. For one thing, I was thinking, what do you name those woodland creatures? Like,
I think
friendly names or?
it's probably too stupid to share.
That's what I was wondering. Is there some sense of humor there? Or are we sticking with like genus, species, or are we actually naming them like Hank?
It's more like the
Or
latter.
yeah, beautiful green, or that yeah. Anyway, and you make skimono, like that's so wonderful. What a lucky kitchen you have, and any of your friends. It's just such a favorite thing of mine, and I've never been able to accomplish that. Although, I have to say, I want to honor and recognize them, the expertise that comes from practice. So, this may be why. So anyway, Yeah, and it was interesting to hear about, to read about your, also your music career, because we're finding so much so that we interview the guests who come here, that many people have had their beginning of their passion, exploring their passion in music. So then I'd like to ask you, um, and knowing a little bit about you, but when did you first recognize that you had an interest in science, and what do you think was that which provided that spark?
Yeah, that's a great question. So I can say that, when I was very little, I was not that interested in science. I was much more into art. Um, and I was always in an imaginary world, so I was not really paying attention in class. I was the sort of kid who was having daydreams all the time. Um, but because my family was moving a lot, my parents wanted to make sure that I didn't get behind in classes. And we were moving, from the US to Japan and back to the US. And they felt that, the one thing that was universal was math. And so they didn't want me to get behind in math. And so that was one thing that, I was somewhat disciplined about doing since childhood. And um, it was kind of natural then for me to go to MIT for my undergrad. I didn't know initially what I wanted to do as a major, but I knew that I didn't want to be a math major, and so I thought I was being slightly rebellious by choosing physics, but it's not that different in the end. But I do think that what drew me to physics initially was the sort of like simplicity and the elegance of like the description of nature that physics provides, and that was really appealing to me. And initially, I wasn't that interested in biology um, or even chemistry, and then it's kind of funny now that that's I do both of those things now. I don't know if I answered your question. I'm sorry.
Yes, absolutely, absolutely you did. You might be the first person who's been in the room who said that they had rebelled through studying physics.
I was like,
That
I'm
is,
not going to be a math major.
yeah, that is cutting edge and wicked. That's great. I love that. Yeah, thank you. So then, can you take us on a little journey up to the work that you're about to present today? Because it is. I think maybe it's one of those things that when we read it, just as humans, we read about what oxygen was toxic to early life, and it takes us aback a little bit, and we have to really consider that, and how did early life get around that. So yeah, so maybe just backing myself up, if you can take us up to the point at which you're doing your research that you're presenting today.
Sure. So you mean like background material to the research,
Just
or
your
the reason
life.
why I ended
Yeah,
up
yeah,
doing this
yeah, yeah.
research,
And
or
and really
okay.
it's your choice. It's guest choice how you choose to answer this. But yeah, just like a you know, not an exhaustive journey, but just things that stuck out to you that you might want to share that brought you up to this point.
Sure. So I can tell you, so when I was a physics major at MIT, initially I didn't know which area of research I was interested in, and I already said that I wasn't interested in biology, to the point that I actually, at MIT, everybody has to take freshman biology, and I delayed it until my senior year because I didn't think that I would enjoy it. Um, but it was actually in a physics class that I first became aware of biological problems that were really interesting to me, and one was the protein folding problem. And up until that point, I thought that biology was, a lot of just memorization. I had this like idea that it was just, about complexity as opposed to sort of breaking down the complexity, which is, what I thought physics was about. And when I learned about the protein folding problem, I thought, well, that is really interesting and really elegant. Then I really became hooked on proteins at that point, and I was probably a sophomore at the time. And the professor who talked about that, his research, and it wasn't, he actually was in polymer physics, but the motivation for his research was in proteins. And that's what got me hooked, and I joined his lab. And more and more I was like becoming enamored by proteins, when it was time to think about graduate school, like I should also mention that being a physics major at MIT, especially the time that I was there, there were very few women. In my graduating year, there were only 11% of us were women. 11% of the physics majors, I mean. So um, I found that, it was really hard to feel like I fit in my class, and the lab provided, like research provided a place where I could belong. And so research became also like really important for me. And then I wanted to go to graduate school to do it more. Um, and oh, one thing that happened actually was my undergraduate advisor really unfortunately passed away. He had a heart attack and he was only 53. And that was totally shocking and really upsetting. But I was so into the research at that point that I didn't want to leave his lab, even though the lab was essentially becoming disbanded. And even though that also meant that I wasn't going to have a letter of recommendation from an undergraduate advisor who knows me well, I just stayed in the lab, and I got permission from the department to keep doing my senior thesis work there. And then when it was time to go to graduate school, sorry, this is like turning into a long story. I'll just say that, I found a mentor who really appreciated that way of thinking about research, and that was really helpful into sort of getting into the field of biophysics and really getting into studying proteins. And then ultimately, though, I felt that in order to really learn about proteins, I had to leave the physics department and go to a department that really cares about molecules. And that's how I ended up in chemistry. And my lab is kind of at the interface of both of these worlds. And we try to develop use physics to develop new methods to sort of study proteins, but ultimately, like, we try to answer questions about proteins. I'm sorry,
Oh,
that was a very long answer.
no, no, please, my goodness, you just given us a gift, and please never apologize for anything any speech like that, or any speech, we mustn't apologize for our presence. And that was a great story of overcoming adversity, too, which is really, it's here we're in Science Society, we're about to hear specifically what what we have in the title, and we never know all of the nonlinear benefits that are coming to us from being here. So that's, just a really great example, and I'm sorry that you had that loss. And you kept going, and also that's very sweet about hearing about a lab that cared about molecules. We got to have more consideration in this world for molecules, and you're clearly a compassionate person. So at this point, I will pass you the mic and just let you know that welcome, Dr. Shaw. Dr. Shaw, Katarina, and I are here to help moderate this space and share all the questions that are coming to you in the room chat after you've given your talk and facilitate a Q&A. So thank you so much, Nozomi, and mic is yours.
Okay, thank you so much. And thanks everyone for coming. This is my first time using the Clubhouse app, and it's I might mess up, but hopefully it will be okay. And so right now, I have my title slide up. So if everybody wants to check out the slides, the title of the talk is Evolution of an Ancient Enzyme. And, I was told beforehand that it's going to be a really diverse audience. If you have any questions in the middle, feel free to interrupt me. What I've prepared is like two sort of short stories in case we don't get to everything, because, I don't want to overload you with information. So we'll just see how far we get. I'll just go to the slide two, which is like, kind of the main story. So I'll tell you about the evolution of this family. I'll introduce the enzyme family and then tell you about the evolution of that, and what we learned about it. This slide is a little outdated because both of these papers have been published now, but it's the work of three very talented graduate students, Audrey and Darren, who are from my lab, and Matt Spence, who is from Colin Jackson's lab at the Australian National University. And this was also a fantastic collaboration that spanned multiple years through a pandemic and wildfires in Australia. So a real triumph. So if we go to slide 3, what you see is on top is what is known as the central dogma of biology. The only thing that I really want you to take away from this is that I'm sure, many of you, most of you have heard of these molecules, right? DNA, RNA, and proteins. These are, major classes of macromolecules, and genetic information usually gets passed from DNA to RNA to proteins. And the building blocks of DNA, so these are all long polymers, and DNA and RNA, the building blocks for them are called deoxyribonucleotides and ribonucleotides. Okay, so these are the individual building blocks that get turned into these long chains that eventually we call DNA and RNA. And what's kind of interesting is that in nature, the way deoxyribonucleotides are made is that it's actually made from ribonucleotides. So when I show this arrow curving to the left, it's showing the direction of the reaction. And the fact that this goes in this direction is really interesting already. And the reason is because there is a hypothesis that, ancient life began with an RNA world. So that RNA was potentially the oldest of these macromolecules. And today, we would consider it a DNA protein world. So the fact that the building blocks of DNA are made from the building blocks of RNA suggests that this chemistry was really important. So we think that based on the sort of ancientness of the sort of hypothesized RNA world, that this chemistry must have been also ancient. And so that's really interesting already. Now, in modern organisms, this chemistry is done by a protein called ribonucleotide reductase, or RNR. So that's the word in blue right there. And because it does this chemistry, already people had sort of proposed that this is an ancient enzyme family that has this transition from the RNA to DNA world would have happened like billions of years ago, by the way. So like really ancient, or at least, it is a descendant of that chemistry. And the enzyme family, the RNR family, is also like biochemically interesting because it uses this, what I'm showing here is a cysteine radical. So cysteine is an amino acid, like one of the 20 regular amino acids in a protein. And it normally has like SH at the very end, but there is like a special cysteine inside the active site, like the most important part of the protein that forms a radical, and that's an unpaired electron, and that's highly reactive. And this is like the sort of thing that ignites chemistry in this protein. And so the fact that this protein uses this kind of chemistry, it's called radical chemistry, is already really interesting from a biochemical standpoint. And this RNR is used by really like every organism that is free living. The only exceptions that we know of are actually some tiny parasites and some viruses that don't need to make DNA, which is kind of weird to think about. A lot of viruses that don't have RNRs, for example, will just like hide the RNRs from the host. So anyways, this is an introduction to that enzyme family. And if we move on to slide 4, I'll give you an idea of how that cysteine radical is made. And so to generate that kind of radical, you need something to help the protein make that radical in the first place. And the way the RNR family does it is through having a cofactor. So cofactor is, could be a small molecule, or it's basically something that is not just a pure protein that is attached to the protein to help it do its chemistry. And it's part of catalysis in the protein. And one cofactor that the RNRs use is this one, adenosylcobalamin. So the molecule on the bottom, this giant molecule on the bottom, that is adenosylcobalamin. The adenylyl part is sort of colored in red, pink. It's that letter A. It's like a shorthand version of that part right, of the chemical structure. And this is actually related to vitamin B12. So this is, I don't know if you know this, but vitamin B12 looks like this. It's pretty wild. If you're, as a physicist, when I came into the chemistry field, I was really like blown away to know that it was such a giant molecule. But anyway, in the way adenosylcobalamin can work to generate a radical is that the bond that is in red, that's a carbon to cobalt bond, can sort of sort of lyse in a way. So it breaks apart in a way that it generates a radical, like an unpaired electron. You can see it as a red dot. And that initial radical can then go on to react with the cysteine, the special cysteine amino acid, and to generate that cysteine radical. So this is one of the cofactors that the RNRs use, but there are actually two other major classes of biochemical cofactors. So if we go to slide 5, you'll see 3 columns, okay? And this is traditionally how the RNRs have been sort of classified or discussed. And it's been discussed in terms of its biochemistry, like how it generates this cysteine radical. And there are three known major classes, so class 1, 2, and 3. And the class 2 one is in the middle. It's in this blue box to indicate that you just need this cofactor inside the protein. So the blue box is the RNR protein, and that's all it needs. It doesn't need another protein. The class 1 and the class 3 RNRs, though, require another protein to generate this cysteine radical. So, for example, in the class 1, it requires another protein that's called a ferritin. That's a type of fold of a protein, and it's really just ferritins are very good at holding metals, and so this complicated like chemical structure in the bottom is showing you sort of amino acids within the ferritin fold that allows it to hold up to 2 metals, M1 and M2, and this can sort of assemble into a cofactor that's attached to the protein and then generate a radical. In this case, on a nearby tyrosine, that's what the X residue is right there, the amino acid that says X equals 2. And then this radical actually has to travel long distance to another protein, to the active site where that special cysteine is. And to do that, it needs to pass the radical onto a stack of other tyrosines. That's what the thing is inside the blue box in the catalytic subunit. The class 3 RNRs use another totally different mechanism, and this uses iron sulfur chemistry. So you can see in the pink box where it says activase, there's like a cube-shaped thing that has irons and sulfurs. That's known as a 4-iron 4-sulfur cluster, and this is a type of cofactor that can assemble under anaerobic conditions. So when there's no oxygen, it can form and be stable. And so the class 3 RNRs uses these clusters ultimately and then the activase sort of interacts with the catalytic subunit to ultimately pass on this radical to another amino acid, which initially goes to a glycine and then to the cysteine radical. So now, if you're not a biochemist, you might say, like, what is the significance about these? So the next thing that I want to talk about is, so if you go to slide 6, is the oxygen sensitivity, which you could see, just make sure that I'm on the right slide, yeah. So you can see that the oxygen sensitivity really depends on the class. So if you look at the class 3 enzymes, iron sulfur clusters are extremely sensitive to oxygen. They will degrade into other forms of clusters of these atoms, or they might just disappear altogether. And this glycine radical that is shown in the blue box that initially forms, that gets passed on to the cysteine radical, is also extremely sensitive to oxygen. And so the class 3 RNRs, you can already get a picture, are used by organisms that live under these anaerobic conditions. The cobalamin cofactor that's used by class 2 RNRs, on the other hand, not particularly, like, especially sensitive to oxygen compared to the class 3 RNRs, and it doesn't require oxygen, so this is kind of like in the middle of 1 and 3. Whereas the class 1 enzymes, you can see in the ferritin subunit, between the 2 metals M1 and M2, there's a bridging oxygen, and that oxygen comes from molecular oxygen. So the class 1 RNRs, in order to do chemistry, is actually dependent on oxygen. So this is really interesting because when you look at these, what's known biochemically about this family, already you get a picture that, you know, these enzymes must have existed for a long time and have adapted somehow to changing levels of oxygen on Earth. So one of the things that, you know, had been hypothesized for a really long time was that, in fact, that it appeared in this order that's shown by this arrow, that the class 3 RNRs, which are oxygen sensitive, appeared first when there was no oxygen on Earth, before oxygenic photosynthesis filled the atmosphere with, you know, a lot of oxygen. And then the class 2 RNRs appeared next, and then when after, you know, photosynthesis had filled the air with a lot of oxygen, then the class 1 RNRs appeared. So this is what people had hypothesized based just purely on sort of biochemistry and thinking about these cofactors. But it's actually a very difficult question because if you think about things like, well, which of these cofactors may have existed in the last universal common ancestor, actually it turns out that cobalamin might have been already there at a time that these iron sulfur clusters were there. So just from these sort of arguments, it's actually really hard to tell the order of evolution. And so for that, you need to do this sort of phylogenetic analysis, which I'll tell you about. But before I get there, right, so this is like the sort of showing the cofactors, and the box, each box here represents a different protein subunit. This is like, you know, when I say subunit, you can just think of it as like, you know, different parts of a protein. If we go to slide 7, though, you'll see what these proteins actually look like. So the class 2 RNRs is structurally the simplest in the sense that you don't need another subunit. So it only has the catalytic subunit. And you could see that you might be able to see that it has some symmetry. So there's the right side and the left side. So there's actually 2 copies usually in most of these RNRs, and it forms something known as a dimer when it's 2 copies. But you can see this like magenta colored stick thing, like this little mess in the middle of each of these sort of protein chains, and that is the cobalamin cofactor. So in contrast, the class 1 RNRs has the ferritin subunit that I told you about. You can see the orange spheres in there. That's where the metals would be normally found, where the radical is first formed, and then that radical has to travel all the way to the catalytic subunit. And if you look inside the sort of like bluish greenish region of each sort of half of the catalytic subunit, you might see like a yellow sphere. That yellow sphere is like the location of the cysteine, the site of the cysteine radical. So that's where the radical has to go to. So it's very long distance. And then the class 3 RNRs, we have the activase protein, which is a separate one. And what you see is like orange and yellow balls. That is the iron sulfur cluster. Okay, so we'll come back to look at these protein structures again, but I just wanted to sort of introduce the chemistry and what that means in terms of the actual proteins. So if we go to slide 7, what I want you to sort of take away with so far is that this ribonucleotide reductase family, the RNR family, you know, has the potential to provide a molecular fossil record of life based on just the cofactor diversity itself, and that it must have spanned sort of it's encoded in the family is the molecular adaptations that happened over a long time and billions of years ago. And that the goal was, by studying the evolution of this family, we learn something about our past, not their past, right? So something about our past on this planet. So to study the evolution of any protein family, though, what you need to do is phylogenetic inference. So that is, if you go to slide 9. So I think many of you have seen, you know, various sort of renderings of a phylogenetic tree, whether it be like very artistic or like a very, you know, like a rigorously made one from like modern bioinformatics methods. A phylogenetic inference is actually like a very computationally extensive way of figuring out how currently existing things came to be. So usually it's done on sequences, either a protein sequence or the gene sequence. And the reason why this is so computationally demanding is because, first of all, you need to have a huge set of sequences that sort of captures the diversity that's sort of in nature right now. And then you have to sort of figure out how the amino acids in these sequences may have mutated to get to this point. And that is really challenging computation. And so the first step of that, this process is, if you go to slide 10, is a multiple sequence alignment. So you take the sequence of, your enzyme of interest, for example, in this case, RNR, right, from all of the sort of organisms where it's been sequenced. And there's much more nuance to this. You have to do some curation of this sequence data set. But the important thing is that, like, the starting point of phylogenetic inferences, so you have to have this matrix of a sequence alignment where you have an idea of like which amino acids should align into a column. And then the whole goal of phylogenetic inference is to use you have models of amino acid substitutions, and sort of thinking about the rates at which this happen in order to be able to relate all of these different sequences together and figure out like when branchings occurred. So like you're going back in time by doing this, but essentially you're kind of building the tree from the ground up in phylogenetic inference. And this has been like a goal of the biochemical field for a long time, but with RNR field, it's been very difficult to do phylogenetic inference. And the reason is because if you go to slide 11, again, now I'm just focusing on the catalytic subunit. This is where the chemistry happens in the RNRs. You can see that they're very different. Just looking at them, you can tell they're very different. So the structural diversity is coming directly from the fact that the sequences are extremely diverse. And you can already tell, that probably even the lengths of the sequences are different, right? And so that means that that initial step of making this multiple sequence alignment is already very difficult for a family of proteins that is this diverse. But if you want to learn about the evolution over such timescales as like billions of years, that's exactly what you have to do. And so our approach to this problem was to focus. So my lab, we do a lot of structural studies. So we think about protein structure a lot. And these sort of examples that I'm showing on slide 11, they look like they're totally different, but actually a structural biologist sees this and sees patterns. And if you go to slide 12, actually, if you focus on the area around this active site, around that sort of special cysteine that I told you, there is actually a motif. And we can call this like the core fold of this protein, and that is conserved. So you can see that if we strip away all of these sort of like things that make the structures different, what's left around this cysteine, the yellow ball, is really similar. And so if we go to slide 13, I can sort of tell you a little bit more about this RNR fold. And this, what I'm showing you on top is a three-dimensional structure, and below is, in the cartoon is called a topology diagram in the field of structural biology. And the topology diagram is a way to think about secondary structure elements. So these arrows represent beta strands, the cylinders represent alpha helices, and the loops are literally connecting loops between these secondary structure elements. And topology diagrams help you sort of think about, like a way, so it's kind of like you can sort of think about the overall structure of something that looks really complicated in 3D, and how, if you were to unfold it and just smush it onto like a piece of paper, how it would look like. So if we look at that topology diagram, you can see that there are actually two halves that look very similar. The first half, and so when we look at a protein sequence, we always start from the N-terminal end to go to the C-terminal end. So the first half is the left side. You see that there is a set of 5 beta strands that are pointing downwards, and that forms a parallel beta sheet that points together. So it forms a wall, the inner wall of this barrel that's shown on the top side in the three-dimensional version. And then on the outside of this wall of this beta sheet that's forming the inner wall is these 4 alpha helices, A, B, C, and D. And the interesting thing about RNR is that there's actually sort of a duplication of that motif on the other side, except now the beta strands are pointing the opposite direction. And this barrel forms in 3 dimensions by kind of wrapping around together such that, the J beta strand will come next to the A beta strand. And then the 2 halves are connected by this yellow loop, which is called the finger loop. And it's called the finger loop because at the very tip of this loop is that special cysteine, which kind of points out like a finger. And this type of fold is actually extremely unusual in the protein field, or like all of proteins that we know of. And this is kind of interesting because there are a lot of motifs that are found that are very common, and like a structural biologist would be able to recognize a lot of motifs that are, for example, deposited in the public protein data bank. But the RNR fold is very unique. It's basically shared by only one other enzyme family that's very much related. So this is kind of interesting. But the reason why I'm telling you about this unique fold is because, now that, we know that there is this unique fold in the RNR family, we can use that as an anchor to figure out how to align the sequences, even if they're extremely diverse, coming from totally unrelated sequences. And so this is how we did sequence alignment, and we called it structure-based because we're using structural information. But it was kind of like an iterative process of sort of increasingly adding more diverse sequences. And then once we did that, we were able to do phylogenetic inference. So the result of that is on slide 14 here. We actually used 2 different sequence evolution models. The top half, you can see it says LG plus R10. That's one type of sequence evolution model. The bottom, it says WAG plus R10. That's another type. The important thing I want you to take away from this is that, phylogenetic inference is very computationally expensive, and we had about 7,000 sequences in our alignment. And because it was very diverse, it was also really, really difficult. Computationally, it took 1.4 million CPU hours to compute this. That was 7 continuous months of supercomputer time. And it actually, in reality, it took us 3 years because we had to do it twice because we noticed a mistake in the first trial. So it's not like an easy feat, but we were very glad to see the result in the end. So when you look at these 20 different replicates, what you notice is that the ones that have a blue sort of cloudy background, they have very similar topologies. So when we say, a topology of a phylogeny, like a phylogenetic tree, we're looking at sort of like how the branchings are occurring. And you can see that there's like red portions, yellow portions, blue portions, and green portions. And the branching is very similar in these blue ones. And that is sort of like the fact that we're reproducing similar topologies is telling us that we've achieved a solution that is reasonable. And another thing that was really sort of promising to us immediately was that what we did here was we actually colored the sequences based on their known biochemical class. So can people still hear me?
Absolutely.
Okay. I'm not used to having like no feedback. So, okay, great.
If you want, I can interrupt you.
Sure, feel free to. um, okay, so I want to explain this color coding on slide 14 for the people who just joined. The red is the class 3, the ones that are sort of based on the sequence. We predict them to be biochemically the class 3 type. The yellow ones are predicted to be class 2 type. The blue ones are class 1 type. Okay, and what you can see is that like the red sort of lines are all kind of coming up from one node, one point. And same with the yellow ones, same with the blue ones. And this is what we call monophyletic clades, so that these sort of groups of these branches are called clades. And what was really promising and exciting to us with this initial result was that, first of all, what is known to be biochemically different is also phylogenetically different. So that means that there was some congruence between computational method and what we understood from biochemistry. But for those who have really good eyes, you might also notice that there's a green portion in all of these. And these are sequences that were known to exist, but people hadn't really thought about them that much in the RNR field. And these green sort of clade, so it forms its own group, so it's a clade, was a clade that had never been noticed before. So if you go to slide 15, you'll see that a little more clearly. This is, we're looking at one of the 20 replicates. And you can see now that the annotation of the biochemical classes 1, 2, and 3, but now we have this sliver of green, the green clade. And those who are familiar with reading phylogenetic tree can see already that this green clade extends very close to the origin of this circle, which is telling us that it's ancestral to something. It is actually like an ancestral clade to the class 1 and 2. And I'll show that again in another slide. But anyway, this was totally unexpected, the fact that there was a phylogenetically distinct clade that had no correlation to sort of like the known major biochemical clades. So if we go to slide 16, on the left is that phylogenetic tree, the same phylogenetic tree that I just showed you, except it's collapsed. So all of the sort of
Wait,
yeah.
Doctor, could you go into the significance of what that means? Like, so people have been ignoring it for a long time, then they come along and they keep finding this. What does it mean?
Exactly. Yeah. So that's exactly what I'm going to get into. Um, I could, so, well, I mean, this, there's a lot of significance. I think the reason why people hadn't noticed that there was a new clade, so if everybody goes back to slide 15, there are multiple reasons. One is that sort of past attempts at doing phylogenetic inferences, people couldn't include all of the sequences. If they were too diverse from what was like known, it made the computation difficult. So either they used a really, really small data set, which excludes things like the green clade that doesn't have that many sequences that are currently known, um, or there was actually one study that included these the sequences in green, except it couldn't include the class 3 RNRs in that as well, because the class 3 RNRs are actually the most diverse. And that was actually, kind of the problem in this RNR field for a long time, is that without having the class 3 RNRs, is like, you can think of it as another anchor, like sort of a way to sort of further think about the evolution of these sequences. Without them, like, it was really hard to get an accurate picture that this green clade was actually its own separate clade. In fact, like, yeah, so this is actually the first time that we could tell that these sequences were different somehow. And in terms of evolutionary significance, I'll get to that next. So if we go to slide 16, on the left we have that same phylogenetic tree, except in the collapsed version. And this is rooted, so-called, at the midpoint. And when it's rooted this way, you can see that the first branch that comes out is the class 3 clade. And then we have the divergence of the class. Oh, sorry, I should say that we named this tiny clade class O. So this is 0 with a sort of dash through it. We're calling it O, just for convenience. And every time, we start with like a branching point and something branches off, that is like a divergence event. So the class O RNRs diverged before the class 1 and 2s diverged from each other. So this is kind of what our phylogenetic inference was telling us. And we had also done a separate type of phylogenetic analysis, a new method called evovelocity analysis. If you're really interested in this, I recommend that you read this paper at the bottom here by he and Yang and Kim. They are so this is like a relatively new paper that came out very recently, and it uses machine learning methods. And what's really interesting about this is that it doesn't use the multiple sequence alignment that a phylogenetic inference requires. So it's really giving us like a complementary view into evolution. And the plot on the right, the way I want you to read this is that every sort of colored dot, and most of them are kind of merged together so you can't tell that they're individual dots, corresponds to a sequence. And what we're seeing is that the class 3 dots are like kind of its own island. And the reason is it's not connected to the other ones is because, it had diverged such a long time ago that it doesn't have a lot of sort of connecting features with the class 1, 2, and O RNRs. But what's really interesting about the right side of this plot is that class 1 and class 2 RNRs sort of share an interface. And at that interface is the class O sequences. So this is kind of telling us the same thing as our phylogenetic inference, which is that the class 3 RNRs are the most diverged. And then the class O RNRs. Do we have a question here?
Yeah, I do. So what do the arrows mean? One second, I'm just going to clip the image. What do the arrows like going around? What do they mean? Like for someone who wasn't really familiar with these types of diagrams.
You can think of it as like the directionality of like the sort of proposed direction of the evolution. So you can think of it as like a flow plot. But you don't have to overthink this. The sort of main thing that I want you to get away from this is that it looks like the class O RNRs have like a really important sort of position in the evolution of the RNRs because it's at the interface between two known biochemical classes, the 1s and the 2s. And this was really interesting because already, I told you that based on biochemistry arguments alone, we would have thought that the class 1 RNRs were the most recently evolved ones because it requires oxygen to make that sort of radical that I told you that is essential for catalysis. So the fact that the class 1 RNRs don't kind of seem to have evolved in parallel with another mechanism, which is the class 2 RNRs, is really interesting. And so the class O RNRs kind of like show that the class 1 and 2 sort of developed in parallel because it's like an ancestor clade to them. So we wanted to understand more about what the class O RNRs look like. So if everybody goes to slide 17, that green clade expanded is what we see on the left here. And you see that, there are a lot of like uncultured phages, but you'll see the word phage and virus a lot. They're both viruses. So phages are just known viruses of bacteria. And what's really fascinating about the class, at least known sequences in the class O clade, is that many of these sequences are associated with cyanophages. So these are viruses that infect cyanobacteria. And cyanobacteria are, for those of you who don't know, the organisms that started oxygenic photosynthesis on Earth. So before plants, cyanobacteria were the ones that sort of innovated oxygenic photosynthesis. And so it's really fascinating that, there are sequences that are associated with the cyanophages because phages would not exist unless the cyanobacteria didn't exist. And there's also a mix of phages as well as bacteria. So all of the circles that are blue as opposed to orange, those are bacterial sequences. And all of the names that are in green, those are sequences that come from genomes that have photosynthetic proteins as well. So the main takeaway from this slide is that the class O clade, whereas like all the other classes, they're sort of representing like all kingdoms of life. The class O clade is really associated with marine organisms and in particular cyanophages. That's really interesting. Okay, so we'll come back to this at the end. If we go to slide 18, one of the first analyses that we did on the class O clade was to just compare them to like known, like biochemically known sequences. So I should have clarified what an all versus all blast is, but you can think of this as doing a sequence alignment, a pairwise sequence alignment. So if you look at every plot, there's 4 of them shown here. So let's just focus on one, such as the Synechococcus phage S CBP4. What this plot means is that we took the class O sequence from this phage, okay, and then we did pairwise sequence alignments with all of the sequences in our data set from the class 1 and the class 2 clades. The class 1 clades in blue and the class 2 clades in yellow, okay? And we see that there is a different shape in these histograms. And this E value is, the lower this E value is, so the more negative it is, that means that the more similar the sequence is. And we can see already that if we just look at the entire group of the class 1 sequences in our data set versus the class 2 sequences, class O sequences resemble class 2. Okay, so just a reminder of what class 1 and 2 are. I showed on the sort of margins of the slide, the class 1 uses a ferritin subunit, whereas the class 2 RNRs use that. They don't have another protein. They just have a cofactor, a cobalamin cofactor that's related to vitamin B12. So this could have been the reason why there was confusion originally in the field of RNR and what these sequences from these class O sequences were. I think that initially people thought that they could be class 2 RNRs and not a sort of distinct class. But if you look even more closely, such as if you go to slide 19, right? So you can't just think about the protein in isolation and think about its function. You also have to see like where it is in the genome of these organisms. So here on the top half, I'm showing the gene neighborhoods of 3 different organisms. And the blue sort of shapes, and those represent the sequence for the RNR catalytic subunit. And in all sequences that were fully sequenced, the genomes that were fully sequenced, we found that there was a ferritin-like gene that's like immediately downstream. So downstream as in like to the right of the catalytic subunit. And that was really interesting because it was annotated as ferritin-like. So you immediately think that maybe it actually requires this ferritin because it's like always together, these 2 genes. And that is more reminiscent of a class 1 RNR. But the reason why it had not been classified definitively as a class 1 RNR is because these ferritin-like genes do not really actually show a lot of similarity to known like bona fide class 1 ferritin subunits. And for those of you who don't know, in the past couple of years, we've seen a lot of advances in structure prediction, and AlphaFold is one of those methods. And AlphaFold, however, is actually able to sort of tell us much more than what sequence alignments can do. And actually, if you take these ferritin-like gene sequences and have AlphaFold predict a model for you, it looks like what's shown here. And that type of structure is very much a ferritin fold. And so this was really fascinating because even though the sequence is class 2-like, this is telling us that the mechanism is probably more class 1-like. And so if we go to slide 20, we're going to zoom in even more into the catalytic subunit, so where the active site is. So we used a method called cryo-electron microscopy, or cryo-EM, and this is a technique that has become really important in recent years, and has given us a way to sort of look at the structure of proteins without making crystals of them. So anyway, we were after a few months of like really characterizing this protein, we were able to get a structure. And what we're seeing here in the outline that's in white is like the cryo-EM map, and built inside it is the sort of atomic model of that. And there are lots of interesting things to say about this structure, but in the interest of time, what I want to focus on is the active site. So in the orange box, and you can see what's like zoomed in, there are 3 amino acids that I want to draw your attention to. The yellow one with the sphere at the end, that is the cysteine, the special cysteine on the finger loop. That's where we get that radical. Ultimately, it's kind of like lighting a candle, right? So that's the candle that we have to light. And next to that cysteine, we have 2 tyrosines, and that has those are the ones with the rings and the red stick at the end. Those are the tyrosine residues. We see 2 tyrosines that are stacked, and we call that like Y is just the abbreviation for a tyrosine. So when there's 2 of them like that, we call that a YY dyad or a tyrosine dyad. And what's really structurally interesting about this is that for any structural biologist in the audience, these 2 amino acids are on the same beta strand, and they're adjacent to each other. So the fact that these amino acids are sort of pointing in the same direction is already unusual structurally. And that is like a hallmark of the class 1 RNRs, because the way radical transfer happens inside this protein to sort of light that candle is through passing the radical from those like red portions of the tyrosine residues to that yellow sphere. And so there's like a path that is like sort of you can see structurally in there. And that means that most likely it uses the same mechanism as the class 1 RNRs, where we have a ferritin subunit that sort of first generates a radical, and then it comes in close proximity to the catalytic subunit to pass this radical all the way to that special cysteine. So this is really interesting. And if we go to slide 21, why this is really interesting is telling us that the class O RNRs probably are oxygen dependent, just like the class 1 RNRs. Okay. So to sort of summarize the structural part of this, if you go to slide 22, so the alpha fold model of that gene that is always associated with the class O RNR looks to be a ferritin, right? So that's the green box, kind of similar to the green box in the class 1 mechanism. And the cryo-EM structure of the catalytic subunit really looks like a class 1 active site, in that it has that stacked tyrosine next to the cysteine. And this is the recipe you need to have this oxygen-dependent mechanism. So based on that, we can sort of think about a model for evolution, and that is slide 23. And this is actually kind of like the end of my first part of my talk, and I think it could be a good place to just end the talk altogether, and we can have a discussion. But so let's sort of piece everything together. So we're on slide 23. The ancestor to the modern RNR, that's the ancestral RNR. That's all the way to the left. Okay, so that's the starting point. We think that it appeared before oxygen was filling the atmosphere on Earth, and the reason is we think that is because we see the sort of like change in the usage of the cofactor necessary to make that radical, right? And we see like a diversity from like very oxygen sensitive all the way to requiring oxygen. So if you sort of align this ancestral planet to the sort of bottom arrow, you see that it emerged during the time when the planet was anoxic, didn't have oxygen. But the presence of the class O RNRs is really significant, because that's telling us that at some point, when the class 3 RNRs diverged, it sort of diverged from the last common ancestor of the aerobic RNR. So the RNRs that were part of organisms that had to adapt to oxygen on Earth. And the fact that the class O RNRs are so associated with cyanophages suggested that they could be very closely associated with the advent of oxygenic photosynthesis, which was innovated by cyanobacteria. And from on this sort of like this branch of the aerobic RNRs, once we had that divergence of the class O RNRs, then we see this the divergence of the class 1 and the 2 RNRs. And what's really cool about this part is that for a long time, we thought that the class 1 RNRs were the most recently evolved. However, what we're seeing now is that this mechanism that uses this ferritin that requires an oxygen to make that radical was already appeared before the class 1 RNRs appeared in the class O RNRs. And so the way to interpret this is that, shortly after, and by shortly, it's probably still like billions of years, right? Not billions, but like on the order of maybe like hundreds of millions of years after oxygen photosynthesis was innovated on the planet, there was enough oxygen locally that organisms had to adapt to that, even though that the planet wasn't filled with with oxygen such that it was like permanently a totally different atmosphere. And so that event is called the Great Oxygenation Event, and that is sort of dated by geochemical records. Like if you think about like the rust layers inside sediments, where you had enough oxygen in the atmosphere that you could sort of form those rust sediments. But before then, there was already oxygen on Earth that sort of promoted or that really like put pressure on life to adapt to it. And this is what I think that the class 1, the RNR family evolution is telling us. So now it's 7 o'clock. I think instead of going on to the next story, I should just take questions and sort of turn this into a discussion.
Well, thank you so much for this amazing talk and for explaining this in such a really, like, a way that even I could understand it. So it was really wonderful. And so just to ask one question, so in the like in the data analysis, like, or in the data that you had from different organisms, were there anaerobic microorganisms in there? And is there some way where you, because you kind of hypothesized that there were locally previously already maybe some oxygen, and they kind of, a few species had to kind of adapt to that, and we have locally regions where there's really no oxygen, and there are even microorganisms that like produce micro oxygen in the dark. We had like a guest speaker here. So do you have data from these in there that could us maybe give clues the other way around, basically? Thank you.
Oh yeah, thanks. Yes, so in the 7,000 sequences in our phylogeny, there are like basically all of the ones that the class 3 RNRs come from are either strict anaerobes, so organisms that really require the lack of oxygen, or facultative anaerobes, so they could switch from maybe like an aerobic to anaerobic metabolism. It's hard to say, like we didn't see like strong patterns though in terms of the organisms and like whether they're strict anaerobes versus facultative anaerobes, and like where they lie in the phylogenetic tree. And part of the reason why I think that happened is because this sort of divergence happened such a long time ago, that we're seeing really like just in modern sequences of the class 3 RNRs. Yeah, so I don't know if I can really answer that question. It basically happened such a long time ago that we can only sort of infer that adaptations happen based on the fact that these new biochemical classes appeared.
Yeah, I can see that. Will it be possible even one day to, like, the original cell, or it's such a long time ago that looks for me that it kind of is almost impossible. But on the other hand, if you have this certain amount of DNA and these molecules available, how many options throughout these hundreds of millions of years can you go through? And probably we just repeat like design or possibilities or mixtures that happened before. Like, do you think that's the case?
That's a really good question. So, I should say, from phylogenetic inference, we are trying to get some information about our past, but we can never really know what happened in reality, right? So these are, sort of still hypotheses based on models that, that we computed. And so we won't know, but one thing that is really cool about doing phylogenetic inference is that you also, in order to figure out how the branchings occurred, you also had to sort of predict what the sequences would have been in the past, and those are called ancestral sequences. And that is something that we are interested in, is looking at these sort of sequence predictions at these branching points, to see, like I personally think that we're not going to see a lot of variability in catalytic mechanisms, by studying these ancestral sequences, but I think we will learn things about, other sort of functional parts of the sort of other sort of functionally important things about the protein, such as like regulatory mechanisms, which I think, didn't have to have appeared like billions of years ago. It's something that, could an organism can still sort of evolve and continue to change.
Yeah, thank you. Rose, please go ahead. Or Dr. Shah also unmuted, so Dr. Shah, go ahead, and then Rose.
Oh,
Okay,
sure.
thank you so much, Nozomi. That was really very fascinating work so far. And my question, you might answer my question later on, but specifically, I was wondering about the immunochemistry and the type of the IHC antibody, because you mentioned about the RRM class one also. Perfectly everything makes sense when you explain about the cobalamin B12, and we can think about the even increasing of the homocysteine. But if you want to explain us about the kind of the marker or antibody which it can help us specifically for the colon tumor, did you think about that, and did you came up with some investigation around it?
Okay, let's see if I can try to understand the question. So, is this more like in terms of disease relevance?
Yeah, because perfectly, if you heard about RRM1 is one of those IHC antibody that they are using for the improvement of the tumor diagnosis for the colon cancer. And when you just explain about all those chemistry behind, it does make sense totally for me. And I was just wondering, when you want to think about the, I mean, medicine, how
Mhm.
you can how you could find it beneficial, or do you have any idea about?
Yeah, that's a really great question. So, RNRs are actually, current drug targets for cancer therapies. And eukaryotes generally use the class 1 RNRs. So, the RNRs that we use don't use the cobalamin cofactor, but so there are actually FDA approved cancer drugs that currently exist, that are being used, for breast cancer and maybe other ones. I think advanced pancreatic cancer, and so maybe not like the best drug, but the reason why though is because RNR is important for cell proliferation, because it's the only way we can get more deoxyribonucleotides to make DNA. And so that is definitely one like area that has been actively studied. But another way in which the RNR family is medically very important is that, um, different pathogens might use a different biochemical mechanism. And so that is, actually, if you're interested, like if you go to slide 30, you'll see some common human pathogens. They use a different type of class 1 RNRs than what humans use. And that is like one of the reasons why, there's interest from the NIH, for example, in understanding, um, the how we might be able to sort of target, pathogenic RNRs without, um, hurting the host, and that would be a different strategy to fighting, infections and that type of disease.
Exactly, because they have a use for that for the fabrication, especially for the viral. and you explained partially. So I'm just passing the mic to the next person, and at the end, if we have time, I might ask more questions. Thank you so much.
Yeah.
So, I'm gonna be annoying for a second and like do a hypothesis. Let's say I'm this uber skeptic person who is skeptical about the idea that this kind of analysis is valid, and ideally, I want to figure out a way to get a sample um, of either bacteria that are somewhat cryopreserved or somehow. I know like our ice sheets only go back like 2 million years or so. This is 2.3 billion years. Is there some way we can actually find like some place we could core, some place where we could find like bacteria that has been cut off from the rest of the earth or are just laying dormant until we come around and like sample them and see how life has changed? Like, is there some place where we can do that? Like, go back billions of years?
That's not an annoying question. I mean, first of all, I mean, I think it's really important to be skeptical about this any type of analysis, but especially something like this because it is really like purely computational. And no offense to people who are just computationalists out there, but I, I think that, like as we get more sequence information and as sort of like maybe theories of evolution continue to improve, we might get like, different answers or better answers. Who knows, right? That question though about like whether we can find sort of like actual fossils that are useful. I mean, I would love to find something like that, right? But I don't know if we'll find anything that is viable that has been really isolated. I don't know. It's possible. There are spores that, have been, I think, revived from being below the earth mantle rocks or something, like some extreme pressure because it's below the rocks below the sea level, that were revived. So maybe, but it's would be really hard, I think, to be able to tell what you're looking at.
So this is gonna sound crazy, but the event when the Chukla crater was formed, I can now pronounce that, the dinosaur event, um, it spewed out a lot of material into space, right? Like that was, if have you read the paper that goes into the timeline of the last day of the dinosaurs? Like one of the events that end up killing most of the life on the planet is that the material after the impact, there's ejecta, and the ejecta goes and spreads, and it goes into some of them. A lot of it is in
Hello,
suborbital trajectories.
everyone, can you hear me?
Yes. Yeah, just give me a second. It's on a suborbital trajectory, and when it re-enters, it starts heating up the Earth's atmosphere, and so much materials re-enters together and so quickly that it raises the Earth's atmosphere for like a minute or so above, or was it 1,000 degrees centigrade? But above, at least above the boiling point of water, right? And it's just basically mass planetary sterilization. So, but some of those rocks also went on an escape trajectory from Earth, and some of them might have even hit the moon. Like, there's these proposals that where they want to track the see when the moon had an atmosphere. So they're gonna do coring in the permanently shadowed craters of the moon to find the ice samples from that time and then evaluate the moon's atmosphere. Maybe we could find like a tiny rock that flew out of that event and is landed on the moon, and it has a sample preserved for all of these billions of years, like hundreds of millions of years. Is
I
something like
mean,
that possible?
I mean, it's possible,
Yeah,
yeah. I mean, it would be amazing.
I just want to say hi. So, are you guys?
I'm sorry. Thank you. I'm sorry. This is a public room. Something stuff like this happen. Just please go
Okay.
on. Thank you.
Gotcha.
I was gonna
Yeah, no,
say,
I mean,
don't you think perhaps that the deep sea sediment cores might have a better, at least a higher chance probably than the dark side of the moon, which seems like the light side of the moon might be better since it's less bombarded in terms
Oh no,
of.
permanently shadowed craters, not the dark side of the moon in the sense we can't see. It's a crater towards the polar ends of the moon. There are craters because of the angle of the sun, where these are permanently cryogenic. Like
Oh,
there no
fascinating.
light ever reaches them, right? So there's ice preserved there from the time when the moon was more still had a magnetic field, it was more active, and still had somewhat of an atmosphere. So, sorry.
Yeah, no, understood. I still think that it might be more likely that we'll find um, some better specimens in deep sea sediments. What do you think of that, Nozomi?
oh, I mean, these are all like really fascinating proposals. I'm, I'm not well versed enough at thinking about the sort of viability of these different sampling locations. I think that I would never discount them as a possibility, and I would personally be fascinated by either. Um, but whether knowing like one is better or the other, I just don't know. Yeah.
That's a great answer. That's a great answer. Like, people need to get better at saying, I, I don't know. Great question.
You're awesome, Dr. Andy. It's and
Thank you. Thanks for the great questions. I mean, it's a really great discussion.
I had a very specific question. So, ferritin in this context still means iron. Is that right?
You think, so iron is a common, but in the RNRs, so in the class 1 RNRs, they all use ferritins, like I told you, but they don't always use iron. Um, there's there are subclasses within the class 1 RNRs that use manganese, like two manganese ions, or a combination of iron and manganese, and so. But it is designed well to hold metals. But yes, irons are sort of easy to sort of get incorporated into the ferritin assembly.
Interesting. Okay, so if I missed this, I apologize, but I'd be really interested to hear your explanation of ferritin because, in chemistry, ferrous metals generally tend to include iron, and then if you were talking about a ferritin level and perhaps like a clinical patient, you'd be looking at iron indicators. But so, could you expand on that?
Oh, interesting. So I wonder, so in a clinical sample, when you say ferritin levels, does it specify like which ferritins?
Great question. I've never had the panel drawn. I just know that patients talk about having um, this assay on them. Dr. Shaw, have you ran a ferritin panel, and could you speak to that at all?
Ferritin level normally is a part of the, I mean, general lab that we are just running on a patient. But specifically what type of ferritin, no, I don't know about that. We are just, I mean, running as it needed. But I just, in a previous question, I asked Nozomi about the B12 and ferritin and cobalamin and how it can be connected with the absorption of the B12, especially when we are talking about IF factor and all of those things. So I think my question was relevant with your question.
Interesting. Yeah, I mean, so Zomi, you're saying that it could be manganese plus iron or just iron. Are there any other configurations, or what else can you tell us about the ferritin phenomena?
Yeah, so I just realized, that there is a protein that people sort of informally called ferritins, that it is a ferritin fold. So when you're doing a ferritin assay, if you actually Google ferritin, or you find it on Wikipedia, you'll see a structure that looks like a ball. And so, in fact, if you want to look at the one on Wikipedia, that ferritin is an iron storage protein, and it is called a ferritin protein. The ferritin that's used by RNRs has a similar fold, but it's not the same protein. So, but they're, probably evolutionarily related in terms of the structure. So it's a
And
sort of,
function, perhaps?
sorry?
And function, perhaps?
Yes, certainly, yeah. So ferritin, when people, maybe like talk about ferritin proteins, I think they're thinking about iron storage. So that could be a good way to think about the iron levels inside of the body. And it is designed well to hold metals, but it can also do sort of actual chemistry as well. And that's what the RNR ferritins are sort of good at.
Wow. And so prior to how many hundred, we were talking about geological timescales before. I think might have been one of the words you were looking for. So this is a phenomena that started how many hundreds of millions of years ago?
That's a really good question. I'm assuming, if the ferritins were being used, uh, you know, like around the time that photosynthesis appeared, that would be like billions of years ago, right? So like a few billion years ago, so this fold would have already existed then. And proteins are really just fascinating though, because you can have um, very different functions for the same fold, right? And even though I think all ferritins are probably good at holding metals, because it's not just the fold, but it's also the specific amino acids that are coming together. If you refer to my slides, you'll see that there's some symmetry as well that helps with the sort of arrangement of the metals. Yeah, but we haven't actually done a phylogenetic analysis of the ferritins themselves. Then I think that would be really interesting too, because they do. So yeah, when I'm saying ferritins, I'm using very loosely the structural family, right? So, but functionally, I think it actually spans many different types of proteins.
Wow, yeah, thank you for teaching that. I was not aware of all these details, at least some of them. So I really appreciate that. And also, I want to leave some room for Coco. Coco will come.
Give me a second. I'm getting dressed.
Yeah. Oh, then
No,
Dennis,
go
did you
ahead. Go
want
ahead.
to say? Oh, I just wanted to check in with Nozomi how much time you still have left, because we've been going for almost an hour and a half. So, I wanted to give you a chance to get some rest. But yeah, yeah, just let us know, and then if it's like one more question or two, and then
One or two sounds good to me. I mean, I'm really enjoying the discussion, but I know that everybody's probably hungry right now, so I don't want to keep you from dinner either.
well, we are fine, but but
Don't worry. People are eating on mute and all these other sorts of.
yeah, you're the only one that has to keep talking. So yeah,
Okay.
let's do like around two questions, and then go from there. So, Dr. Shah, did you wanna ask the other question you had in mind, or?
I might wait till the end if there is, I mean, time for that, because I'm interested to see that what is coming at the conclusion.
Yeah, thank you so much. And based on your work, would you also be able to make like predictions for life on other planets, maybe how life on Mars would have looked like? Do you think, RNRs would be important everywhere in the universe for life, or do you think like we cannot really make predictions like that on other, planets, because we don't know enough about the environment?
Yeah, I think, well, I mean, I love this question. I think that, the honest answer, though, is that I think it is very difficult. Um, I don't know if I mean, this RNR is really only necessary for DNA-based life. And there, I think, are, I'm sure, other mechanisms for doing the same chemistry, and it's just that on Earth, this is the way it works, and this is the way that nature decided it works. It's going to keep using it, and it doesn't want to lose this protein and so badly, so that it's just going to keep adapting, so that we can use it in different environments. But I think I don't know. I mean, I can say that this was a type of project that I've been wanting to do for like over a decade, because I've been sort of enamored by this enzyme family for so long, and having done the sort of analysis, I realized just how complicated it really is. And so there are actually not that many studies done to this level of like complicated enzyme families. So a good place to really start is just doing more of these studies, so that we can sort of start thinking about predictions, right? I think we're still kind of at the basic science stage, where we're trying to understand more. Does that make sense?
Oh yeah, totally. Yeah, thank you for that answer. And oh, Rose, you had that question, so please go ahead.
Oh, I was just gonna ask, if you had like $20 million in supercomputing credit, what would you do? Like, would you run this? A genuine question, right? Like, if tomorrow, one of these large foundations, like the Simmons Foundation, like the Flatiron Institute, etc.,
Uh-huh.
they go like, "Hey, we'll give you $20 million of supercomputer time. Uh, what do you want? Like, what is the way forward here?"
Well, I mean, that is a really great question. Also, you guys, I love how big you guys are thinking. um, I think there are a lot of evolutionary questions that I'm still interested in. And obviously that would really help to have computational time. So there are other enzyme families that will shed light onto not only how like the early planet may have sort of been, as well as learning the connection between like how does life adapt at the sort of protein level to changing environments, and all of that. I think obviously like having more computational time would really help, because just considering just how long it took to just do this one. But my lab doesn't just do evolution, so we actually the reason why we got into this is because we were interested in studying uh, understanding what makes proteins seem alive, in the sense that coming from a structural perspective, right? So if you see these pictures of protein structures, and they look very static, but we all know that it's not static, it's dynamic, it's always moving, every molecule is moving. But macromolecules, I think, are able to adopt really rich motions that are essential for its function, for its chemistry, and also regulating that chemistry. And we don't really understand how that works. And this is when people talk about proteins, they almost seem like they're alive somehow, because they're just able to do these amazing functions. And really, what my lab would like to be able to do is to understand that the molecular mechanisms that sort of make proteins seem alive, and that to me is like trying to understand the relationship between this protein sequence and protein three-dimensional structure, and how it moves, and then the function, right? And so connecting all of that is really like one of the ultimate goals of my field. And thinking about the sort of dynamic part is really difficult. So that's another thing that I would spend money on. So like the evolution part is really interesting to me, just because I'm really interested in the past, but it's also like a way to sort of rationally change protein sequence. And so it's one way to sort of change sequence, and then see how that changes all the other things. Um, but that's really just kind of getting at like part of what we really want to know ultimately. So the sort of thinking about dynamics requires a lot of computation as well. So yeah, that maybe that the way that I would answer it is I would do like 10 million in protein dynamics and 10 million in evolutionary studies. And but I do think that like if we really that there are separate goals and common goals between the two halves, but fundamentally, if we can understand, why these macromolecules, these proteins, are able to do these amazing functions with, moving beyond these static pictures, that would be like really amazing.
That's an excellent answer. Like, I like thinking about agent-based kind of simulations to see, hey, how do these pieces come about? What's connecting them? Why do they exist? And like that kind of thing. Like, what is the simulation that we're thinking about?
Um, well, all sorts. So, because, my lab is kind of at the interface of sort of physics and biochemistry, the physics side, what we're trying to do is think about like developing methods that allow us to sort of think about or visualize these protein motions. And that's traditionally very difficult to do because in structural biology, the way you get a clear picture is to have all of them be identical to each other. And that is not going to be possible if they're moving, right? So if they're jiggling around, they're not going to look like they're identical. But this like sort of, in order to get enough signal, that averaging is necessary for like all structural techniques. And so it's always kind of like a trade-off of like getting an insight into motion means that we're probably going to lose some other information about like higher resolution. So like how can we sort of tease apart the information from these moving molecules and sort of interpret that, and that that requires sort of like more data. So some of the computation is like being able to crunch the data, and some of the computation is just like thinking about new software that needs to be developed to be able to analyze the data. And then there's computation that needs to be done in terms of like simulating these sort of motions and then seeing if it agrees with experiment. And there's like an entire field of molecular dynamics. And of course, that's kind of like what we want is something like that, except we want it to be absolutely realistic. And that we have not achieved that yet because that's really difficult, especially if we want these simulations to be as accurate, say, as an experiment. So in experiments, we can put error bars on things, and that precision is actually extremely small for certain techniques. But we're not there yet with computational techniques. So like sort of bridging that gap would also be really important.
Thank you so much for entertaining me.
Okay.
Yeah, thank you so much for those questions and the answer. It's really interesting. We could go on and talk about what means life and where does it start. Do you think, those proteins, all molecules that, change around, is it a very simple form of life? But that's maybe questions for your next, when you come back here next year someday or something. If you would honor us again, that would be amazing.
Yeah, thank you so much for the great discussion and for tuning in.
Yeah, no, thank you for coming and giving this amazing talk and answering our questions. And we really appreciate you taking the time to do this. And we wish you all the best. And maybe you get this grant from the Summons Foundation. actually, no, a friend of mine is working there at the computational part there.
See? See? It's possible.
Yeah. So, well, yeah. So all the luck, and I hope you get all the grants you want. And yeah, congratulations for this wonderful work and life. Like you seem to, you seem to figure out how to find time for different passions, which is amazing. So congratulations on that too.
Thank you. I'm not sure if I'm doing a great job at that, but I appreciate it.
Well, Well, great. We are life. We are always improving. Nothing is perfect. So as long as we try. Okay. Yeah. Enjoy the rest of your evening. Thank you, everyone, for coming. Enjoy the rest of your morning, day, evening, wherever you are. We appreciate you coming. And if you like discussions like this, follow the club, follow our guest speaker, and our moderators. Tomorrow we'll have a talk with Dr. Xi, and he is using also omics data integration to develop personalized drug discoveries. So yeah, I'm sorry, it's on Thursday, not tomorrow. It's on Thursday. So yeah, thank you so much, Zumi, again. This was wonderful. It was such a treat. And thank you, everyone.
All right, thank you, everyone.
Yeah, close the room in 3, 2, 1. Bye, everyone. Thank you.